Kate, I have a question for you. How do you approach training and standardization of processes across the different manufacturing sites, for example, or clinical
Mhm.
centers for thawed product handling?
I think that's a really good question. And it touches on a lot of what was just said, where, you know, I think it's really, really critical to develop good training programs. And usually what I found interesting is you develop a training program that is sort of, this giant comprehensive program, and then you actually figure out how to stream it down to the smallest program possible because it needs to be simple to be taught and it needs to be streamlined to be accessible. And one of the things that we have found, and I think that many have found, is that when you go to multiple clinical sites, part of it is what works in your BSc or GMP or whatever setting that you're in for cell thaw, while it's going to work in another setting as well, the setup of the room might be different. The setup of reagents might be different. The accessibility of certain equipment might be different. And so really having your staff that are experts go in and train those facilities and making sure that you are tailoring anything that needs to be adapted for that facility, even the timing of certain testing coming back, how testing is sent out, if it needs to be sent out at all, making sure that you're really working and understanding what the particulars are that site has going. Because I think we are at the point where there are many, many skilled cell therapy groups at really excellent clinical centers, but they all may work a little bit differently. So making sure that your training program or your SOPs actually translate to their environment rather than trying to force something that isn't exactly going to work where they are is really critical. So, we like to do on-site assessments, make sure that our quality team is available, make sure that our SMEs are available for things like dose prep, for thaw, for processing, and really go in and work with the centers, because even when a group, and rightfully so, will tell you, we've done so many thaw procedures, we have no problem, it's so easy for us. And that is absolutely true. They are incredibly skilled, but your process is always a little bit different from somebody else's process, and figuring out which step that may be challenging for them is absolutely critical, or which step could be adapted slightly to their facility without an impact to the process, because we know our process extremely well, is important. And I think getting constant feedback from those teams and from the sites really helps you even further refine that process as you go. So I think, even speaking to things like not implementing automation too early, because some of those things may or may not work across different facilities. I think that's really important. And even refining your training materials as you go and you see how different facilities work. So I think that that's been an incredible learning experience to understand what is absolutely required versus what do we think is required because our facility works that way.
Right.
So yeah, that's sort of just what we've seen as we've expanded, especially from phase 1 to multi-center phase 2s.
Excellent. Good. Okay. Thank you. I want to move on to talk about cell quality and consistency. So, if we're going to attempt to really kind of tackle the IPSC cryopreservation, we want to make sure we're ensuring quality, consistency, scalability. So, I'd like to go through some of the critical challenges and perhaps innovative solutions in preserving IPSCs for next generation allogeneic therapies. Pratik, I'd love to start with you. How do we ensure consistent quality across different batches of IPSCs during cryopreservation?
Yeah, good question. So really it starts from the very beginning. When we start developing, well, maybe I'll start with the idea that our formulations, we have developed a toolbox of different formulation kind of baselines. And so what we have is that as we understand a particular cell type that we're working with, we can leverage our toolbox to kind of help us understand more subtle properties of those cell lines or those cell types in particular. And then we start tailor making our formulations to kind of match the performance that we need to see. So if we're talking about something like recovery, post-thaw recovery, well, if we know that one particular cell type is more sensitive to shear forces, then we might try to minimize any type of mechanical forces that we might have in how we might conduct a harvest or some type of cell concentration. So try to build that into our overall workflow from the very beginning, and once we have a sense of it. And then over time, what we'll do is we'll also try to increase the number of runs that we do to better understand how exactly are we seeing it from batch to batch to batch. And we start leveraging in-process analytics to help define what we would define as kind of release characteristics. And so that way we're matching pre and post any unit operation activity, and we're kind of monitoring performance. So maybe just in a general sense, you can think of all the things that we've already talked about, things like cell counting and identity testing, some type of functional test if you have one. The idea there is you're trying to see how those are changing over time. There's other subtle tests you can also do, things like looking at cell surface markers. You can look at metabolism, just to see subtle differences and shifts that you might project if there is indeed an impact on your processing steps. So that's the way that we've been thinking about it. And we build that data set so that we get closer and closer to, let's say, an IND package or IND ready package. We have quite a few N numbers to be able to rely back on and say, this is a consistency that we've seen. And then we also define some targets for ourselves. So what are we aiming for? And we kind of measure ourselves against that.
Love it. Okay, great. Before I move on to my next question, I just certainly want to offer up to Kate and or Bruno to provide some color commentary on that as well, about ensuring consistent quality across different batches of IPSCs during cryo.
I can jump in really quickly and just say,
Sure.
as if cell therapy wasn't complicated enough, right? We tell us that there's cell to cell variability, cell type to cell type, there's batch to batch variability. And honestly, the other thing that I don't know that we've talked about here is just even touching on lot to lot reagent variability that you can run into even in the GMP setting. So being able to have metrics, critical quality attributes, things that you can measure for all of these components, I think is really critical. I also think, just building on what Pratik said, is know your cell type. Cells, different cell lines, different cell types behave differently. And so really being masters of the biology of your cell guides you in what to look at. Do you need to look at post-thaw cell recovery? Do you need to look at activity? Do you need to look at composition? I think that really understanding what you're working with. So for example, NKs and gamma deltas, they might both be classified as innate immune cells. They behave very, very differently in regards to cryopreservation and recovery from thaw. And so I think that understanding what you need to look at based on your cell type is critical. I think hopefully getting down some batch to batch variability as you go through your PD and through your development, that generally happens as a byproduct of that. But one thing that we've also noted is lot to lot variability in reagents, and making sure you have a good program to qualify your reagents when they come in. Even when you expect GMP grade not to have that, that can pop up from time to time, and that can really impact the quality of your cells if those reagents do differ. So making sure that you have in-house ways to look at those, or standard ways to look at those reagents, we think is incredibly valuable, because I think oftentimes when you see something unexpected and you chase it down, that can be the root cause of a lot of it. Can be the reagents, disposables, something that you don't expect to massively impact the process that does. And then I'm sure I'll pass it off to Bruno, because I'm sure that you've also seen a lot of these things.
Yeah, no, thanks, Kate. I just wanted to maybe highlight a couple of things that you and Pratik mentioned. One is establishing a pre-cryo baseline is absolutely critical, right? And then as Kate was just mentioning, that's going to vary the specific assays, depending on your cell type and your process and whatnot. The other thing that I wanted to mention, and maybe this becomes a fun part of this conversation, and maybe it doesn't, but I find it pretty interesting, and I guess I'll say it in a half joking way, is post-cryo, how hard do you look, right? Because right after cryo, you have a choice, right? Do you start assaying your cells immediately after they come out of thaw? Do you wait 24 hours? Do you wait 48 hours? Do you wait 72 hours as the quality of your cells presumably, right, deteriorates, right, as the effects of cryo are more pronounced? Now, where do you stop, right? And then similar to that, what assays do you look, right? Do you just use a regular old viability assay like Trypan Blue, right, which is not highly sensitive, and there, your cryo process tends to be excellent. But the more that you have more sensitive assays, let's say an Annexin-5 assay that, gives you information about cell health, live cells, apoptotic cells, the picture changes, considerably. So I think that those are some of the fun considerations or the interesting considerations for this. But, so set the pre-cryo baseline. It's absolutely critical. And then as Pratik mentioning, having a good understanding of what your goalpost is post-cryo, really makes the challenge a lot more doable, or else you end up, chasing ghosts a little bit,
Thank you, Bruno. I want to get to, as I'm taking a peek at our questions from our audience, which we will get to later in the hour. It turns out the next question I wanted to go through with you would be about the innovative storage solutions that support scalability requirements of allogeneic therapies. So, you know, what are those innovative solutions and how are they supporting the scalability requirements of allo therapies, considering the need for storing large batches of cells? I'll offer it up to any one of you, but perhaps Pratik, can I start with you?
Yeah, maybe I didn't quite understand the question itself. I mean, when you're talking about innovative, or when you're asking about innovative solutions, is that, you know, for individual settings? Is it for distribution, long-term storage? Because there's lots of different options, and the field is evolving. But maybe I can just answer as I think I understood the question. Unless Aaron, you want to add some more detail?
Nope, have at it. Go ahead.
Okay. So, I mean, there's been a lot of innovation in the space on how we think about, let's say, liquid nitrogen dewars. There's other types of smart dewars that will allow you to be able to transport a certain number of cells in a vial format, bag format, across distances. And of course, that's very important. The more recent technologies have allowed those systems to be GPS enabled. It also allows you to have real-time tracking. There's other elements around the length of time for which they're validated. So you can actually have, rather than hours or just a couple of days, potentially, you know, maybe a week or more is what you can have in terms of being able to ship. But maybe long-term storage in a facility of some kind, that probably still looks very much the same as it has before. It's usually these large LN2 storage units. Maybe the technology is improving in maybe how you think of being able to retrieve a particular vial. That's something where, if you think of it like a vending machine, the more advanced technologies that you can see that are a bit pricey but still are being adopted slowly, basically allow you to be able to get a single vial or a single set of vials rather than opening up the entire freezer, pulling out a box as you might have done traditionally, and getting exactly what you need. So you're not impacting or disturbing anything else. That allows you to have that temperature control much longer. So there are some innovations that are happening in that space. But of course, there's an investment here in terms of money and dollars to be able to get trained on it, to have it installed. There's also some footprint requirements. Some of these systems, especially the vending machine type systems, require a larger footprint than traditional LN2 freezers. So maybe that's how I interpreted the question. Maybe Bruno and Kate have other thoughts here.
Feel free to take it on. I know both of you are, again, agreeing, so I certainly know you agree with Pratik, but feel free to tackle the question as well.
I think one of the things that comes up, you know, more and more, given some of the extreme weather we've seen, and one thing that is worth considering is, where do you store and how do you diversify or spread out your risk through multiple storage sites? I think that's a discussion that many companies have had. You know, there are some traditional areas where there's been a lot of storage, you know, Houston, Florida, areas that may be considered geographically risky at this point. You know, but every time you move product, even though you're using LN2 shippers, even though you often have a week in that type of shipping solution for cryo, every time you move something, there is a risk to it. So how do you balance the risk of wanting to have multiple storage facilities so that you never fully go down in case there is something that impacts one facility? And then how do you distribute product in a way that minimizes the risk to your company because you are moving product? And then the thing that everyone has to consider is, then when you are treating patients, how do you get your product to those clinical sites? And you know, get it from wherever you're storing to the clinical site. How do you validate all those lanes? Do you want to validate multiple lanes from multiple storage sites? Do you just want one lane to a site for your product so that you don't have to have that extra cost? So, you know, there are so many considerations in not just how you freeze and when you freeze in your process, but where you store. And so I think that most companies have gotten to the model of having at least 2 offsite storage facilities. But, you know, again, that's a risk, and all of those things also come with cost. So there's a lot of work to do on the front end to actually balance how you're storing on the back end. So I think running those risk analyses, those discussions early on is really helpful as a company to understand where you're going. So you don't all of a sudden realize that you've built an infrastructure that is just not sustainable or that's not practical for actually running a clinical trial.
Good, thank you, Kate. Bruno, I want to offer you up the opportunity before we move fully into controlled rate freezer operations.
Sure. So once again, I agree completely with Pratik and Kate, and again, just offering my maybe somewhat ancillary perspective here, which is, as we expand our allogeneic cell therapy clinical trials across the globe, right? So going from a US market, let's say, to an European market. Interesting, maybe not only storage related issues, but logistical issues, in that a number of cell therapy regulators are still very much used to autologous therapy, right? And how we have handled chain of custody, and then they see that this is a cell therapy, and they don't really lock in that it's allogeneic, and they have certain expectations that are very much related to autologous. So I'll give you an example. Having the subject ID on the label of each vial, right, is a regulatory requirement in some places. And how do we do that for an allogeneic therapy, right, where the point is to serve many, many patients with a single batch, right? So just wanted to kind of offer that up as an example. It's not, related specifically to storage solutions, Aaron, but something that the field is going to need to contend with as we move from autologous to allogeneic cell therapies.